to our presentation on DNA replication.
Let us begin by talking about why DNA replication is important. The information stored in DNA is essential for life. If a cell dies, the body must replace the cell. The only way to replace the cell is to replicate the information that the cell contained. When cells die, DNA helps replicate them to keep us healthy, growing, and alive. For example, if you got in a fist fight, and since it is the winter, your knuckles cracked. Because of this, you need to repair the millions of cells that were killed when you cut your fists. DNA replicates those cells lost in the fight. DNA replication is also necessary for reproduction. Parents give their children DNA when they replicate to make cells in order for them to grow into an adult. A third reason is cancer cells kill healthy cells. DNA replication helps replace those cells with new healthy cells.
In order to begin the complex process that is replication, the double helix must first be unzipped into single strands, otherwise known as the replication fork. In order to do so, the hydrogen bonds between the purines and pyrimidines must be broken. The pyrimidines include cytosine and thymine, and the purines include adenine and guanine. Note that there are 2 hydrogen bonds that exist between adenine and thymine, and 3 between cytosine and guanine. A and T can only match with each other, and C and G can only match with each other. DNA is directional and signified by a 5' and 3' end. Whether it is a 5' or 3' end tells us whether a phosphate group or a hydroxyl group is attached. In the case of the 5' end, phosphates will be attached, and for the 3' end, hydroxyls will be attached. Additionally, because of this difference in direction, the processes of replication differ slightly. The enzyme that performs this process is known as DNA helicase. This direction is important because in replication, the fork creates 2 strands. The leading strand is one oriented in the 3' to 5' direction, while the lagging strand is 5' to 3'. This directional change is called antiparallel.
A short piece of RNA, otherwise known as the RNA primer, binds to the 3' end of the leading strand. The primer will always bind to the starting point, similar to makeup primer, in how we put it on at the start to make sure all of their makeup stays on. These primers are generated by an enzyme called DNA primase.
In the next step, DNA polymerases make new strands. This process is known as elongation. Replication proceeds in the 5' to 3' direction on the leading strand, so the newly formed strand is continuous.
The replication of the lagging strand is slightly different. It begins replication when the RNA primer lays down the matching nitrogenous bases. The DNA polymerase then lays down nitrogenous bases between the primers the RNA laid down. These sections are called Okazaki fragments. Having many different fragments creates a discontinuous process of replication. The reason for this discontinuation is because the Okazaki fragments are disjointed.
The DNA polymerase then returns and changes the RNA sections to DNA.
After the continuous and discontinuous replications are complete, an enzyme known as DNA ligase joins Okazaki fragments together to form a single strand.
DNA replication is an endergonic process because it uses energy or ATP to begin. Additionally, throughout the process, the different enzymes require energy to aid in catalyzing the reaction. The energy needed to create a growing strand comes from the cutting of high-energy phosphate bonds on the nucleoside triphosphates.